Another way DNA can move through bacteria is through transformation. Discovered by Fred Griffith in 1928, transformation is a cell's uptake of a donor bacterium's DNA from the surroundings. It maintains this DNA so it can pass it down to its daughter cells. The process begins when the bacterium lyses and releases its DNA. If the fragments come into contact with a cell that is able to take up DNA and transform, or a competent cell, then it will bind to it and be taken in by the cell. The DNA will then be integrated into the recipient's genes and be transformed. What makes a cell competent? Cell competency depends on several conditions, such as stage of growth. Once a population becomes competent, they secrete a protein called competence factor. This protein stimulates the production of 8 to 10 new proteins required for transformation. Some bacteria are naturally competent, meaning they don't need to be doused with calcium chloride or undergo electric shock to be able to take in DNA. One such bacterium is Streptococcus pneumoniae. For S. pneumoniae to integrate a donor bacterium's DNA, the DNA must first bind to a cell surface receptor. It then cleaves it to double-stranded fragments about 5,000 to 15,000 base pairs in size using an extracellular endonuclease. To integrate the double-stranded DNA, one strand must be hydrolyzed by an envelope-associated exonuclease during uptake. This will enable the other strand, now single-stranded, to align with the homologous region of the recipient's genome and be integrated into the chromosome via homologous recombination. What takes the DNA inside? Protein complexes. They have the task of moving DNA through the bacterial cell wall. In the gram-negative bacterium, Neisseria gonorrhoeae, there are 5 proteins involved in the complex. The first is the protein PIL-Q. It aids in the movement across the outer membrane. Then the piline complex PIL-E moves the DNA through the periplasm and peptidoglycan. The COM-E protein binds the DNA, then protein N, a nuclease, degrades one strand before the DNA enters the cytoplasm through the membrane channel formed by COM-A. On the other hand, the protein complex of the gram-positive bacterium, Bacillus subtilis, is localized to the poles of the cell, meaning it cannot go outside the peptidoglycan layer. Most of the proteins involved in its complex are similar to N. gonorrhoeae. The piline complex, COM-GC, PIL-E in Neisseria, DNA binding protein COM-EA, COM-E in Neisseria, nuclease N, and the channel protein COM-EC, COM-A in Neisseria. A uniquely gram-positive protein is COM-FA, a DNA translocase that moves DNA into the cytoplasm. Application. In research, geneticists use transformation to move recombinant DNA into cells. Some of the bacteria that are used in the experiments are not naturally competent, such as E. coli. Thus, they need to undergo special treatments such as electric shock and exposure to calcium chloride. Both methods are used to make E. coli more permeable to DNA, making it artificially competent. To summarize, in transformation, 2 different cell types are used: artificially competent cells and naturally competent cells. On one hand, artificially competent cells is a cell type that requires 2 factors, electric shock and calcium chloride, to be able to take in DNA from the donor bacterium. On the other hand, naturally competent cells is the type that does not require electric shock and calcium chloride to be able to take in DNA. It has its natural competence factor, which would naturally stimulate the synthesis of a certain protein, which would be the one to take in DNA from the donor bacterium. The process begins when a bacterium lyses and releases its DNA. If the fragments come into contact with a cell that is able to take up DNA and transform, or a competent cell, then it will bind to it and be taken in by the cell. The DNA will then be integrated into the recipient's genes and be transformed.